whenever we talk about mutations, we are making reference to changes that take place in the sequence of nucleotides, a section of DNA that pertains to a gene. Remember, we've learned in class that a gene is going to be the sequence of DNA bases responsible for spelling the instructions on how to build a polypeptide. We don't call mutations changes in chromosome structure, and we don't call a mutation if a change happens during the transcription of DNA to messenger RNA. If the messenger RNA is changed in any way, that would not be characterized as a mutation. Mutation is more of a permanent nature if the change in the sequence of bases doesn't get fixed right away. What leads to mutations will be changes, agents such as radiation. And you may ask me, well, what kind of radiation? Think about forms of energy in the environment, such as UV rays that come from the sun. You can also think about X-rays and a few other forms of radiation that we may be exposed to as a consequence of our lifestyle or due to medical treatments. So another thing is going to be chemicals. Many times petroleum derivatives are going to contain chemicals that if they enter the cell and make it all the way to the nucleus, can alter the sequence of bases in DNA. And so overall, there's going to be two types of mutations. One type of mutation is just what we call nucleotide substitution or base substitution. So in a gene, everything is going to look right, everything is going to appear to be correct, except that there may be just one base that gets switched for an incorrect one. So if you look at this example on this slide, you can see that in normal hemoglobin, there's going to be a codon in DNA that is specifically the bases C, T, and T. When this codon is copied to messenger RNA, the C goes with G, the T goes with A, the T goes with A. This mRNA codon can be found in our genetic dictionary. And if you take the time to find it in your textbook or another source where you have this genetic dictionary, you will find that this is going to be translated as the amino acid glutamate. And so this amino acid is needed for hemoglobin to fold in the globular shape that it needs to be in order to transport oxygen and carbon dioxide as well. But on people with sickle cell disease, a mutation that happened, we don't know exactly when or where in the African continent, this middle T was substituted for an incorrect base, which in this case happens to be A. So the codon in DNA CAT gets transcribed as GUA, but the translation of GUA is not the amino acid we expect, but instead it's going to be valine. And just this one incorrect amino acid is going to result in the hemoglobin polypeptide to not fold in the way it should. Consequently, the red blood cells that are made with these abnormally folded hemoglobin polypeptides are not going to be performing the right function because they don't have the right shape. And so this type of mutation where a base is substituted is also known as a point mutation. And we can see in this illustration that point mutations sometimes can be silent. Sometimes they don't have to cause a particular harmful effect. And this is because of the redundancy of the genetic code. Sometimes we have up to 3 or 4 different codons that can call for the same amino acid. And so in the example you see provided in this illustration, you can see that there is a base substitution. There should be G, G, and C calling for glycine. If the last C is changed for U, the G, G, and U still calls for glycine. And so because you still end up with the normal chain of amino acids, there is no consequences in the phenotype. There is no genetic disorder or defect. And so we call this type of pair substitution a silent mutation because it leaves no effects. Sometimes the substitution of a base in the case of the GGC, the first G is changed for an A, and this calls for the amino acid serine. This one amino acid is incorrect. Look again on this illustration on the normal gene, which we call the wild type. There should be glycine right before stop, but instead we end up with serine before stop. And so this type of a change leaves a consequence, and the consequence is described as a missense. Missense is going to be one incorrect amino acid, and this has the potential for a polypeptide not to fold in the right way, not to have the right shape, and consequently to not function. If the base substitution happens earlier, for example, as we have the second codon here, AAG, now let's look at the mutated DNA. We have UAG. The A has been replaced by U. This is going to be calling for a stop codon. It's going to be terminating the assembly of the polypeptide, and when the polypeptide assembly is terminated prematurely, we call this type of a consequence a nonsense effect. And so notice that the effects of these base substitutions can be categorized as silent, if there is no consequence to the phenotype because the right polypeptide still forms. Missense means that one of the amino acids is incorrect, and nonsense means that there is a premature stop codon that is terminating too early the assembly of a polypeptide. Another type of mutation we can see is what we call frameshift mutations. And a frameshift mutation can happen if a base is substituted or if a base is deleted. So once again in this illustration, you can see a normal gene. This would be like the wild type we saw before, where the amino acid methionine, remember methionine is always the first amino acid. This is what the start codon is asking for, followed by lysine, phenylalanine, glycine, and then alanine. If, however, there is one of the bases that gets removed, in this case, one of the three U's is taken out. What it does is that it pulls all of the other bases up one location, changing the frame in which these codons are going to be read. If every other base after the deletion, after the removal of one base, moves up one location, the new codons will be UUG, which calls for leucine, GCG, which calls for alanine, and CAU, which calls for histidine. So notice that these three amino acids following the deletion of one of the bases are going to be completely different from how things were in the original gene or the wild type. Base insertions will also be a frameshift mutation, because if we add an extra base, then every other base is going to be moving up one position, changing how the codons are read, and consequently there's going to be highly abnormal polypeptides or proteins. So if I go back to a previous illustration, you can see here what happens, for example, when a base is going to be inserted or deleted. And so in this case, there's going to be an extra U that is added. This is going to be a base insertion, and this moves up all of the other bases, changing the reading frame for the codons, and in this case is asking for, or is calling for, the termination of the assembly of the polypeptide. There is a premature stop codon, and as we learned before, this is called a nonsense. In this case, you can see in the description, the effect is going to be an immediate nonsense, and this will be a polypeptide that will not be able to function. In this case, the case that follows, we can see when a base is going to be removed. This is similar to the illustration I showed you previously to this one. And if you have like a whole sequence of many amino acids that is incorrect, we're going to call this extensive missense. So what's the difference between missense and extensive missense? So missense alone is just when one single amino acid is incorrect. But if you have more than one amino acid, many amino acids that are incorrectly assembled in this polypeptide, then we're going to be calling this extensive missense. So the key things to remember here is that there are going to be two types of mutations. The ones when a base is substituted for an incorrect one, these are also known as point mutations. You can see on this table the consequences: silent, missense, nonsense, or they can also be frameshift mutations, and these happen when bases are deleted or bases are inserted. And these are going to be detrimental mutations because they will usually result in the building of a polypeptide that will not function, will not be folding the right way. And again, on this illustration, you can see that there are going to be consequences like the early stop codon, the nonsense, or extensive missense as well. What is the outcome of all of these mutations? Well, the outcome is going to be the development of cancer. Cancer is a genetic disease. Mutations that lead to an increased rate of cell division are going to be those that lead to the development of tumors and eventually the development of cancer. Cancer cells have abnormal cytoplasms like the one you see in this illustration. Notice that the plasma membrane and the internal cytoskeleton are forming projections that are not typically seen in most cells. There's going to be an abnormal metabolism, meaning that these cells are using a lot of energy. There's a fast rate of division. Depending on the size of origin, cancers can be classified as carcinomas. If it's a lining tissue, like an epidermal tissue inside of the body or outside of the body, sarcomas are going to be cancers that originate in bones or muscle. Lymphomas are cancers that originate in lymphatic nodes. Cancers can also be classified as primary or secondary. A primary cancer is the one that forms in the tissue where the mutations took place, but if the cancer spreads to other parts of the body, those are going to be known as secondary cancer. So sometimes a cancer may begin as a cancer of the prostate, but if those cells spread to other parts of the body, they may also end up causing tumors and leading to liver cancer or cancer of the pancreas. Notice that the types of mutations are going to be determining also the effect on the body. The genes that are susceptible to mutations are going to be known as proto-oncogenes. Now, proto-oncogene is a gene that functions normally controlling the production of growth factors or something like the cyclin-dependent kinases. When a proto-oncogene, which remember is a good gene, becomes mutated, then it becomes an oncogene. And the oncogene this is the term doctors use to refer to a cancer-causing gene. So when a proto-oncogene goes bad, it becomes an oncogene. So oncogenes sometimes can lead to a hyperactive protein that is stimulating cell division, like a cyclin that is not breaking down when it should, and it continues to promote cell division. That can be the case here. Or maybe there is an excess of something like a growth factor that is stimulating cells to divide. Then that's because sometimes there's like multiple copies of a gene in a chromosome, or sometimes you move a gene to a new location in a chromosome, and that is what causes the excess of growth stimulating proteins. The other part of cancer is going to be the tumor suppressor genes. And again, when you think about a tumor suppressor genes, think about it like the brakes, what is halting, what is preventing cells from dividing. So tumor suppressor gene is a good thing. But if a tumor suppressor gene is mutated, it may lose its ability to inhibit cell division. It's like losing the brakes in your car. And without the brakes, then this inactive tumor suppressor gene will be allowing for cell division to continue out of control, and cells will be dividing, violating the inhibitions we talked about previously, like density-dependent inhibition. Cells will be clumping up where they are not needed, or they may also begin to form in places where they were not originally from, so they can detach, move to other parts of the body, and cause trouble. Cancer is said to be something that requires many mutations. So just one mutation will not lead to cancer. One mutation can lead to an increase in cell division. This happens when an oncogene is activated. Remember, these oncogenes will be the ones that stimulate increased cell division. Eventually, a polyp can begin to form, and that's usually linked to a second mutation. And this is going to be when a tumor suppressor gene it's inactivated. One of those famous tumor suppressor genes in our bodies is known as P23. And a malignant cancer or a malignant tumor is going to be one that has the potential to spread to other parts of the body, and that usually takes place when a second tumor suppressor gene becomes inactivated. So some cancers are said to require, 4 to 6 different mutations before a cell becomes malignant and has the potential for spreading to other parts of the body. And this is why doctors want to operate right away as soon as a tumor is detected, to prevent those cells from continuing to accumulate mutations that can eventually lead to metastasis, the spreading of a cancer. Here's a table that is definitely outdated, but it shows you some of the most common types of cancers. It is outdated because now the most common form of cancer in the United States is lung cancer, followed by cancer of the prostate, and then we have breast cancer. But I like the table in that it still shows some of the risk factors, things that a person may have as part of their lifestyle, and may contribute to the development of mutations, and again, potentially to the development of cancer. That is all I have to say with regards to mutations, and I hope that you find this explanation helpful.